Okay, today we're going to talk about somatic mosaicism as a general phenomenon and in terms of its origins, but it's particularly relevant to cancer. So cancer results from mutations that occur during the life of an individual. So in that sense, cancer is a genetic disease because it results from mutations that deactivate important regulatory pathways in our cells. So cancer cells do differ from normal cells in various ways, and it has to do with cellular behavior and how they behave relative to the regulatory signals that they're supposed to see and respond to. So mutations, we have the general class of proteins called tumor suppressors, and what those generally do is restrain the cell cycle, preventing growth from occurring under inappropriate conditions. Those mutations are generally recessive, but do become homozygous during the lifetime of the individual. Mutations that activate proto-oncogenes lead to cell cycle proliferation under inappropriate conditions as well, but these are generally not inherited. It takes a lot of mutations and a lot of genetic events. No single mutation will do it, but we have a lot of cells and we live a long time. So the cells of the body are not identical, even though they all arose from mitotic division from a single fertilized egg. And the reason, of course, is that mutations and genetic changes can occur during the many, many cell divisions that arise when the fertilized egg grows and develops into a multicellular organism. So mutations of various types, including gains and losses of whole chromosomes and part of chromosomes, can occur in various cell lineages as we grow and develop. So here's a picture representing not cancer specifically, but what can happen during the development of an organism. We're starting off with a fertilized egg, and the first division is shown to give you two cells. Well, as those cells continue to proliferate, they can experience mutations, and once they do, those mutations are then passed on to the progeny of those cells, creating clonal populations of cells which contain same mutations as each other, but are also subject to the acquisition of even more mutations. Now, some mutations lead to cell lethality, and those cells do not lead to clonal populations. Others can revert to wild type as well. So many different things can occur with the idea that all the cells of your body have different genetic histories, and so we have subpopulations of our cells with different mutations in them. Okay, so the progeny that contain the mutation leads us to being mosaic in the sense that we have patchworks of cells having different genetic changes that have been acquired during our lifetimes. Okay, so these are somatic mutations that are not passed to our children. Only mutations that occur in the germline are passed to our children, but they can have very great effect on our own lives. Okay, so the face of the mutant cell, sometimes the cells can die. Sometimes they continue to proliferate normal and there's no adverse effects at all. They can revert, but sometimes they proliferate excessively, and that's where issues arise. Okay, so here's one way of representing cancer as it is caused by a succession of mutations occurring during the life of the person. So the purple little dots say there's a simple initiating mutation that's occurred, but as that cell continues to divide, it can acquire a second mutation and a subpopulation, and then a third and a fourth eventually, until the cell acquires a significant number of mutations that lead to excess proliferation and the formation of a tumor. Okay, so this idea is a little more graphically shown in this particular slide, where we have a normal cell and its clonal progeny, and then the first mutation and its clonal progeny. But along the path, that cell, one of its progeny, acquires a second mutation, and then a third, and then a fourth. So it's the acquisition of multiple mutations within a cell population that eventually gives rise to the malignant cells that form the tumor. So types of changes, basically anything you can think of. You can have just mutation of a usual base pair or a few base pairs. The more significant one has to do with mitotic nondisjunction and mitotic recombination, which lead to loss of heterozygosity. Now, why that's significant will become clear in a minute, but generally gains or loss of parts of chromosomes can also occur as a result of DNA strand breakage and other processes. So the two ones we want to talk about a little bit today are the RB protein, which is retinoblastoma. It's the job of the RB protein to prevent cell cycle entry when nutrients are not sufficient. So I'll talk about the role of that specifically in a second, but mutations that inactivate this gene lead to cell cycle entry when this is not appropriate. The second one is P53, which is a mutant protein in Fraumeni syndrome. Okay, this protein is involved in several mechanisms that respond to DNA damage especially. So when that occurs, you're supposed to have programmed cell death if the damage is too serious. This particular protein encodes a transcription factor that leads to cell cycle arrest and that leads to the production of DNA repair enzymes. So it's really central to the DNA damage response, and so it's essential, and it's called the guardian of the genome for very good reason. So these are both tumor suppressors because their absence leads to predisposition to cancer. Okay, so retinoblastoma is the most famous inherited cancer syndrome, being the earliest to be understood. It's an eye tumor that results from lack of functional RB protein in certain cells. Okay, Fraumeni syndrome leads to many types of cancer. Okay, now the inherited part about this is something that's a little bit hard to understand because people that have this Fraumeni syndrome actually inherited only one mutant copy of the gene, not two. So even though they're born heterozygous for the wild type and mutant alleles, they still inherit the propensity to get cancer as appearing as an autosomal dominant. So it turns out that a phenomenon known as loss of heterozygosity is what leads to the development of cancer. Okay, so here's a retinoblastoma pedigree. The people that are colored red have the disease, and the normal ones are not. And it does segregate as an autosomal dominant in the sense that people inherit it from affected parents. Okay, so what the RB protein does in brief is that what it's supposed to do is restrain the cell cycle. So early in the cell cycle, it binds to an important transcriptional regulator, maintaining it in the inactive state. So its job is to restrain the activity of this transcriptional regulator. Well, the job of this particular transcriptional regulator is to promote cell cycle entry by activating genes that are involved in DNA replication. It's restrained early in the cell cycle to prevent those genes from getting transcribed when it's not time to do so. So in a normal cell cycle, when the nutrient signaling pathways say things are good, the RB protein is inactivated by phosphorylation, at which point it loses its restraining characteristics and lets the transcription factor loose so that it can then promote DNA replication by transcribing genes that are required for that process. So you can see that if you didn't have RB in the first place, you don't need to have the signals to cause it to be released from the transcription factor. And the transcription factor can go ahead and just be active, irrespective of the position in the cell cycle. So this is going to drive cell proliferation under very inappropriate circumstances. Okay, so its job is to prevent cell division if nutritional conditions are not right. So in its absence, cell proliferation can continue. Okay, here's a Fraumeni syndrome pedigree. Again, it appears to be a dominant. People with this particular disease have cancer at very high frequency and at particularly young ages. For example, even in this one pedigree, we have gynecological cancer, breast cancer, leukemia cancer, brain tumor, breast cancer, another brain tumor, liposarcoma, colon cancer, and leukemia, all within this one family, and at relatively young ages in most cases. Okay, so this is an inherited predisposition to cancer. It's not like they're inheriting their cancer. They're certainly not, but they're having a high likelihood of developing cancer in their life. Now, not everybody that is born with a P53 mutation necessarily gets cancer, and even if they do, it could be at different times in their life. So the roles that this protein plays in restraining the cell cycle in response to DNA damage explains why or how that particular protein prevents cancer. Okay, so here's a slightly intimidating diagram. All we need to look at here is the p53 protein, which is sitting here in the middle of this complicated series of interactions. Okay, the point is when there's DNA damage, p53 becomes activated, and when that happens, it plays roles in three very central processes. One is apoptosis, the other is G1 and G2 arrest, and the other is DNA repair. Okay, so apoptosis is a process whereby if the cell DNA is badly damaged, the cell basically commits cell suicide and self-destructs. So that's an important way of getting rid of damaged DNA if the DNA is if it's so bad that it cannot be repaired. The other thing it does is contribute to maintaining G1 and G2 arrest in the sense of not allowing the cell cycle to continue until the DNA damage has been fixed. So it's an very important process whereby the cell, for example, will not go into mitosis if it has broken chromosomes or broken DNA, and there are pathways that assure that that occurs, and p53 is an important part of those pathways. Similarly, if you've got DNA damage, you need to repair it, and p53 is a transcription factor that increases the transcription and therefore the protein amounts of proteins that are involved in DNA repair. So p53 stimulates DNA repair by inducing the transcription of genes that are involved in that process. So clearly p53 is very central to our protection from DNA damage to prevent cells with damaged DNA from going into productive cell cycles. Okay, so it's called the guardian of the genome for the obvious reasons that the cell cycle must be made to stop if the DNA is damaged, triggers cell death, and it also is a transcription factor that induces genes for DNA repair. So it's clearly a very important protein. Okay, so the pedigrees show an autosomal dominant form of the gene, meaning that inheriting a single mutant allele is enough to cause predisposition to cancer. Okay, now the idea of penetrance here is going to become important later. Not everybody that inherits the mutant gene actually gets cancer. It causes a predisposition, but does not itself directly cause cancer. Okay, so the important thing to note here is that people that are inheriting the RB early Fraumeni, in fact BRCA1 mutations, those mutations are recessive in the single cells. Like everybody has one normal copy and one mutant copy at the time they're born, and yet the people end up getting cancer. Okay, so the idea here is what happens is that during development, during life, genetic events occur in those cells which cause loss of heterozygosity, or which is to say, cause the mutant form to become homozygous in some of their cells. So even though all their cells have one wild type and one mutant copy, some of them become homozygous for the mutant copy, and those are the cells that are going to then give rise to the tumor. Okay, so they're called tumor suppressors because their normal role is to prevent cells from growing abnormally. So the basis of the cancer is loss of function, and that the protein is not able to carry out this function as it used to. So we need a second mutation to cause this to really be a problem, because they are in fact recessive as single cells, but they do become homozygous during development or during life of the person. Okay, recessive at the cellular level, dominant at the organismal level. Predisposition is inherited as a dominant Y. Events during life lead to loss of heterozygosity, and those are the cells that form the tumor. Okay, so mechanisms: mitotic nondisjunction, mitotic recombination, mutation of the previous wild-type allele, DNA damage, chromosome loss, and many other mechanisms can contribute or can provide the mechanism for a previously heterozygous gene to become homozygous now. Okay, so here's mitotic nondisjunction. What's supposed to happen during mitosis is the yellow chromosome and the blue chromosome are homologs, and each one is supposed to separate from its sister chromatid so that each daughter cell gets one yellow chromatid and one blue chromatid. So the sister chromatids separate, go to separate poles in mitosis, and that maintains the genotype of the original cell. Sometimes you have nondisjunction. We heard about nondisjunction in the sense of meiosis, but it can occur in mitosis as well. And in this particular example, the yellow chromosome sister chromatids did not separate correctly, so that both of them were segregated to the same cell. Okay. Major serious important one is mitotic recombination, which leads to loss of heterozygosity. Now this is not a frequent event, but it's not unheard of either. Okay, if you look at these chromosomes, they look like they're paired. They're not. This is mitosis. We're not pairing and recombining here. But the recombination, which is to say exchange of parts of chromatids, can occur during mitosis, especially in cases where there's DNA damage. So what's being shown here is we have the two members of the homologous pair. One, the red chromosome contains the normal RB allele, the wild type. The green chromosome contains the mutant allele. The first thing that happens when you're getting ready to divide is you replicate your chromatids. So after S phase is complete, we have two red chromatids on one chromosome, two green chromatids on the other one. So what can happen is at that phase, when replication has already occurred, is that the red and the green chromosomes exchange part of their parts. So in this particular example, we've taken a little bit of the chromosome from the green one and swapped it over, so now it's attached to the red centromere, and vice versa. This is a reciprocal event in this case. So now the green chromosome is now itself heterozygous. It actually contains the wild type allele, because that's what's present on the red chromosome, as well as the mutant allele on the other chromatid. Similarly, the red chromosome now contains one copy of the mutant allele as well as the wild type allele. So that's not itself a problem. We're still heterozygous. What happens is when the problem occurs when we segregate these chromosomes at mitosis, and it depends on what pattern of mitosis those chromatids take. So sometimes you retain heterozygosity, and sometimes you lose heterozygosity, according to which chromosome or which chromatid segregates with which chromatid from the other chromosome. Okay, so for example, let's take the red chromatid. So if the red chromatid, which is not the recombinant one, the red chromatid segregates with the green chromatid, right? So we have four chromatids here. The unrecombined ones that segregates. The other chromosome would therefore carry the swapped one, right? So depending on what happens, like if you have a situation where you've got the red and the green normally, that's still heterozygous because you still have one wild type and one mutant allele. And the reciprocal product, one of the reciprocal products, contains the green mutant, but also the wild type. So depending on which ones go where, those are still perfectly normal. The problem arises here when you have the red chromatid segregating with the green one that has a wild type allele on it. Okay, so we were supposed to have one mutant and one wild type go to each cell. Here we have the red one carrying the normal wild type, and then the swapped version going to the same cell. Well, if that's happening, what's next is that the green chromatid with the mutation is also segregating with the red chromatid with the mutation. So that particular combination of chromatid segregation at mitosis gives you a cell and a population of cells after it that are actually now homozygous for that mutation. So it's not the exchange of material that caused the problem, it's the way those chromatids segregated so that both of the mutant alleles ended up in the same cell and its progeny. So those cells, the ones that got the green mutant and the ones that got the red mutant, are going to lack functional RB, and those are now homozygous for that mutation. Okay, so here's another way of showing what can happen to lead to loss of heterozygosity. Okay, we're showing in the germline this person is heterozygous. They have one mutant allele and one wild type allele, right? In this particular case, the wild type allele is carried on the chromosome that's colored white. The wild type rather. The mutant allele is carried on the red chromosome. So what happens during life at some point, that what they're showing by the arrow is simply a deletion or a mutation of some sort in the region of the white chromosome that carries the wild type allele. So that wild type allele is gone now, and that's not able to then protect the person from absence of this protein. So now we're fully mutant. We have a red chromosome with a mutation on it, and a white chromosome with nothing. So the mutation wins, there's only one copy, and it's mutant, and that leads to the possibility of forming a tumor. And they're showing here also the fact that when you do their genotyping, the person, most of their cells are actually heterozygous, but in the tumor itself, there is no wild-type allele. You just have the mutant allele. Okay. So this is just in summary, really, that the tumor, the causes a tumor of the eye, the genetic predisposition, etc. Okay, and the idea we are born heterozygous, but homozygosity occurs during life, and when that happens, tumor can develop in the progeny of the cells that acquired that mutation. So RB is an example of a tumor suppressor. Okay, this is depicted here in showing a relationship to the so-called sporadic forms of the disease. So a way of looking at this is that the original zygote, the person, was born with a wild-type copy and a mutant copy. So they're heterozygous for the mutation, so they should be fine. But what happens then during their life is either there's a second mutation or a mitotic crossover leading to loss of heterozygosity, so that the cells that derive from those events have only the mutant form of the gene. They don't have any wild type to protect them the way the zygote did. So there's more than one mechanism for generating homozygosity, and this basically represents two of these. Now, in contrast to the sporadic form, which is not inherited, it actually takes two independent mutations or events to cause the problem. So what happens is that the people without the pre-existing mutation can still get the disease, but what happens is they have a mutation and then a second event in order to homozygose it. So this is relatively rare, but it does happen, and that is the source of the so-called sporadic, non-inherited forms of this disease. Okay, blood is drawn from a child who has retinoblastoma. If the normal allele is plus and the mutant form is minus, we expect the genotype of the blood cells to be plus 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 minus minus minus. We think they're plus minus because most of the cells of the child are heterozygous, since that is his germline, or the genotype he was born with. The cell sample is taken from the child's tumor, and the DNA is analyzed for the presence or absence of the mutant allele. We expect the genotype to be plus 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 minus 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 minus. The tumor contains the cells that have lost the wild-type allele by one mechanism or another and are now homozygous for the mutant form of the gene and therefore subject to the development of the disease. Okay, look at worksheet. We're going to thank the core source in the Genetics Society of America for the pedigrees, which look at cancer syndromes and how they behave in families, specifically cancers that are predominantly found in males or females, and how that plays out in terms of pedigrees. Okay. One is going to be early onset breast cancer, which is caused by mutations of the BRCA1 gene. Now, males do not get early onset breast cancer as a result of this mutation, but that doesn't mean they can't inherit the mutant gene. They can pass the mutant gene to their children, especially their female children, who will then manifest the disease. So the males don't get the disease, but they can pass it along. Okay, some things about these pedigrees, though. As usual, people from the outside are assumed to be normal for both alleles, right? They may not necessarily always be, but in general that's true because of the relative rarity of these things. Okay, so here's your classic BRCA1 pedigree. The women get ovarian and breast cancer at high frequency and at relatively young ages. Okay, BRCA1 is an important protein involving many mechanisms for protecting the cell against unwanted cell proliferation, especially when DNA damage is present. It's an important central protein in many of these processes. Okay, so this is the BRCA1 gene pedigree. Individuals 1-1 and 1-4 are assumed to be plus plus, which is to say they carry only the wild type alleles. Also, these mutations are sufficiently rare that minus people are not seen or born at in terms of their regular genotype. Now, you might homozygose it during your life, but you're not born as minus minus. So you can write in the genotypes of people as far as that can be determined, if we've got plus plus, and etc. Okay, where it gets more interesting is that the people that are asymptomatic carriers, okay, nobody's minus minus, everybody's either plus plus or plus minus. Okay, some people are going to be asymptomatic carriers because they are male. Males will more likely not show the disease, but will be able to pass down the mutant allele to their children. Right, so in that pedigree, 1-3 is male. He will not get early onset breast cancer, but he did pass the trait to his daughter. Okay, is 2-2 at high risk for breast cancer? I would say yes, because her sister has the disease. She may have inherited the defective gene, but not yet developed it, because we have penetrance issues too. Some people will never develop it, but others will develop it, and the age at which they develop it may vary. So she may not have developed it yet, but she may still. Or she could have inherited the wild type allele. We just can't tell. Here's a prostate cancer. Okay, this is the other situation where males, of course, can get prostate cancer, whereas females who do not have prostate glands cannot get prostate cancer. However, there are genes that are involved, if mutated, lead to a predisposition to prostate cancer. And if a female inherits it, she can then pass it on to her children without getting the disease herself. So in this pedigree, we have 1-3, who's a male with prostate cancer, and he passes it on to his son. His two daughters, 2-4, 2-5, each have a 50-50 chance of having inherited that mutant allele. If so, they can pass it to their sons. And they can pass the mutant allele to their children, and their sons may be at risk for prostate cancer. Here we have a case, 1-2, a female who does not have prostate cancer, but did apparently pass the mutant allele to her son, who does have prostate cancer. So she's an asymptomatic carrier because she passed it to her son without actually showing the disease herself. Her two children, 2-1 and 2-2, you can't tell about them. They each have a 50-50 chance of having gotten the mutant allele, but until they have children of their own or get prostate cancer, you can't really tell what their genotypes are. Okay. So try to assign genotypes to as many people in the pedigree as you can, and there are going to be some where you say, I can't tell, because you just don't know yet. So here's another disease of similar sort. Affects only females. Males can be carriers. And you can see in the pedigree we have 1, 2, 3, 4, 5, 6 females that have the trait. Again, we assume people marrying from outside of the family are plus plus, but that leaves 2-2 as an asymptomatic carrier because he is not going to get this particular disease because it's restricted to females, but he can pass the trait along to his children, and in fact 3 of his 4 daughters do have this particular disease. Now 1-1 and 1-2 are hard to say. One of them carries the mutant allele, not both, either one and potentially, but so you can't really say who is the person passing this down. It's more likely to be the male because he wouldn't show the disease in any case, but it could, from what we know, be either the male or female that's passing this trait along to their children. Okay, which person above is definitely an asymptomatic carrier? 2-1, 2-2, 2-5. Answer is B, 2-2. Wrong sex. Okay, look at 1-1 and 1-2. It's true that the man is a carrier, the woman is a carrier. Either the man or the woman could be a carrier. So either one could, more likely the man. Okay, again, you should be able to assign genotypes in this pedigree and write probably if the genotype is uncertain and state your reasoning.